If we consider the top 5 boons science has gifted humanity during the last century, vaccine would definitely top the list. This is not just a topic of current debate due to the ongoing pandemic, but during the last 10 or 20 years, there has been many doubts and apprehensions among the general public, and thus gave birth to many anti-vaccine campaigners across the globe. Pale Blue Thoughts have released 2 videos earlier related to vaccines and immunity. We really thought a lot before making this video, as our previous video on vaccine trials did not get much views. We then thought, let's go ahead and give our viewers the complete details about vaccines, and hence this 2-part video titled A to V of vaccines. Why A to V? Well, it's because it covers almost all that you would need to know about vaccines, but we, not being medical professionals, we can't really say A to Z. But the details have been taken from reputed medical journals like PubMed and WHO. So, what are you waiting for? Let's take the first dose of the video.
A vaccine is a biological preparation that provides active acquired immunity to a particular infectious disease. A vaccine typically contains an agent that resembles a disease-causing microorganism, and it's often made from weakened or killed form of the microbe, its toxins, or one of the surface proteins. The agent stimulates the body's immune system to recognize the agent as a threat, destroy it, and to further recognize and destroy any of the microorganisms associated with that agent that it may encounter in the future. Vaccines can be prophylactic, which means to prevent or improve the effects of a future infection by a natural pathogen, or therapeutic, to fight a disease that has already occurred, such as influenza.
The first disease that people tried to prevent by inoculation was most likely smallpox. Smallpox was a devastating disease. On average, 3 out of 10 people who got it died. Those who survived were usually left with scars which were sometimes severe. One of the first methods for controlling the spread of smallpox was the use of variolation. Named after the virus that causes smallpox, the variola virus. Variolation is the process by which material from smallpox sores or pustules were given to people who had never had smallpox. This was done either by scratching the material into the arm or inhaling it through the nose. With both types of variolation, people usually went on to develop the symptoms associated with smallpox, such as fever and a rash. However, fewer people died from variolation than if they had acquired smallpox naturally. The basis for vaccination began in 1796 when an English doctor named Edward Jenner observed that milkmaids who had gotten cowpox did not show any symptoms of smallpox after variolation. The first experiment to test this theory involved a milkmaid named Sarah Nelms and James Phipps, the 9-year-old son of Jenner's gardener. Dr. Jenner took material from a cowpox sore on Nelm's hand and inoculated it into Phipps' arm. Months later, Jenner exposed Phipps a number of times to the variola virus, but Phipps never developed smallpox. More experiments followed, and in 1801, Jenner published his treatise on the origin of the vaccine inoculation, in which he summarized his discoveries and expressed hope that the annihilation of the smallpox, the most dreadful scourge of the human species, must be the final result of this practice. The immunization was called vaccination because it was derived from a virus affecting cows.
Louis Pasteur furthered the concept through his work in microbiology. Louis Pasteur was a French biologist, microbiologist, and a chemist famous for his discoveries of the principles of vaccination and pasteurization. Pasteur's first important discovery in the study of vaccination came in 1879 and concerned a disease called chicken cholera. He inoculated chickens with the attenuated form and demonstrated that the chickens were resistant to the fully virulent strain. From then on, Pasteur directed all his experimental work toward the problem of immunization and applied this principle to many other diseases.
Vaccines are made by taking viruses or bacteria and weakening them so that they can't reproduce or replicate themselves very well, or made in such a way so that they can't replicate at all. Children who have been given vaccines are exposed to enough of the virus or bacteria to develop immunity, but not enough to make them sick. There are 4 ways that viruses and bacteria are weakened to make vaccines.
Change the virus blueprints or genes that the virus replicates poorly. This is how the measles, mumps, rubella and varicella vaccines are made. The virus blueprint is changed by a technique called cell culture adaptation, adapting a virus to grow in specialized cells grown in the lab instead of cells it normally grows in. Because viruses can still, to some extent, make copies of themselves after cell culture adaptation, they are often referred to as live, attenuated or weakened viruses. Destroy the virus blueprint or genes so that the virus can't replicate at all. This is how the killed polio vaccine or polio shot is made. Vaccine virus is made by treating polio virus with the chemical formaldehyde. This treatment permanently destroys the polio genes so that the virus can no longer replicate, using only a part of the virus or bacteria. This is how the hepatitis B and in part pertussis or whooping cough vaccines are made. Because the viral or bacterial genes are not present in the vaccine, the viruses or bacteria can't replicate. Take the toxin that is released from the bacteria, purify it and kill it so that it can't do any harm. Some bacteria cause disease not by replicating but by manufacturing harmful proteins called toxins. For example, bacteria like diphtheria, tetanus and pertussis all caused disease by producing toxins. To make the vaccines against these bacteria, toxins are purified and killed with chemicals such as formaldehyde. Again, because bacterial genes are not part of the vaccine, bacteria can't replicate.
A vaccine works by training the immune system to recognize and combat pathogens, either viruses or bacteria. To do this, certain molecules from the pathogen must be introduced into the body to trigger an immune response. These molecules are called antigens and they are present in all viruses and bacteria. By injecting these antigens into the body, the immune system can safely learn to recognize them as hostile invaders, thereby producing antibodies and, most importantly, remember them for the future. If the bacteria or virus reappears, the immune system will recognize the antigens immediately and attack aggressively well before the pathogen can spread and cause sickness. Vaccines don't just work on an individual level, they protect entire populations. Once enough people are immunized, opportunities for an outbreak of disease become so low that even people who aren't immunized benefit. Essentially, a bacteria or virus simply won't have enough eligible hosts to establish a foothold and will eventually die out entirely. This phenomenon is called herd immunity or community immunity, and it has allowed once devastating diseases to be eliminated entirely without needing to vaccinate every individual. This is critical because there will always be a percentage of the population that cannot be vaccinated. Thanks to herd immunity, these people are kept safe because diseases are never given a chance to spread through a population.
Live attenuated vaccines. For these type of vaccines, a weaker asymptomatic form of the virus or bacteria is introduced into the body. Because it is weakened, the pathogen will not spread and cause sickness. But the immune system will still learn to recognize its antigens and know how to fight in the future. Inactivated vaccines. For these vaccines, the specific virus or bacteria is killed with heat or chemicals, and its dead cells are then introduced into the body. Even though the pathogen is dead, the immune system can still learn from its antigens how to fight live versions of it in the future. Toxoid vaccines. Some bacterial diseases damage the body by secreting harmful chemicals or toxins. For these bacteria, scientists are able to deactivate some of the toxins using a mixture of formaldehyde and water. These dead toxins are then safely injected into the body. The immune system learns well enough from the dead toxins to fight off living toxins should they ever make an appearance. Conjugate vaccines. Some bacteria possess an outer coating of sugar molecules. This camouflage their antigens and fool young immune systems. To get around this problem, scientists can link an antigen from another recognizable pathogen to the sugary coating of the camouflaged bacteria. And as a result, the body's immune system learns to recognize the sugary camouflage itself as harmful and immediately attacks it and its carrier if it enters the body. For some diseases, scientists are able to isolate a specific protein or carbohydrate from the pathogen that, when injected into the body, can train the immune system to react without provoking sickness. DNA vaccines. DNA vaccines would dispense with all unnecessary parts of the bacteria or virus and instead contain just an injection of a few parts of the pathogen's DNA. These DNA strands would instruct the immune system to produce antigens for combating the pathogen all by itself. As a result, these vaccines would be very efficient immune system trainers. They are also cheap and easy to produce. DNA vaccines for influenza and herpes are currently in human testing phase. The COVID-19 Pfizer and Moderna are mRNA vaccines or messenger RNA vaccines.
So now that you have a fair bit of understanding of the history and methods of vaccination, we will pause here in the interest of time, and we'll also give you some time to assimilate the information that we have shared. In our next video, we will talk about the possible negative aspects of vaccines as projected by anti-vaxxers, and we'll look at what triggered this worldwide belief that vaccines were bad and harmful. Keep your comments and suggestions coming in, and don't forget to share this video with your friends and family. Until next time, it's bye-bye from Pale Blue Thoughts.